Hello life science students, this is a continuation of the genetic central dogma video. So this takes further what we learned about replication, and we're going to learn about the other parts of the central dogma. Haven't really gotten into it yet, but we're going to talk about translation and transcription. So while I was away, Mr. Francis started this analogy. The DNA is like a cookbook, and I really, really like that analogy. So we're going to kind of run with that analogy. So, if DNA is like a cookbook, then just like you, and I think actually Mr. Francis went into this, if you went to the library, you looked at a cookbook, you saw a recipe that you wanted to try, you would take that recipe by either photocopying it or writing it down on an index card, and that would then represent a piece of the cookbook. So just part of it is this recipe that you take home with you. So you take this recipe home with you, and then you, as a chef, have to use that recipe to put together ingredients to make a pizza or whatever food that the recipe calls for. So let's take this back to the DNA analogy thing. The cookbook is like the DNA. When you make a representation, it's not an exact copy, but something pretty darn similar, something that represents a snippet of the DNA, that's what we call RNA. Well, in this case, it's going to be your mRNA. That part of it is mRNA. So the snippet representation of the DNA is mRNA. And then in the cell, there is a specific part of the cell that reads the mRNA, and that's called the ribosome. So the ribosome reads the mRNA, and while it's reading the mRNA, it gathers the ingredients, and these ingredients are specific molecules called amino acids. So it takes these amino acids and gathers them and organizes them in a specific way that's defined by the mRNA to make your final product, which is protein. So this analogy just works perfectly. And this is the central dogma. So the central dogma is the process of starting with DNA, which can be replicated, starting with DNA, making a representation of the DNA, a portion of the DNA, and that is the first step of the central dogma, which is transcription. And then it takes that part of the DNA, takes that representation of a part of the DNA to the ribosome, so outside of the nucleus, to the ribosome, assembles amino acids in a specific way to make proteins. And that part is called translation, which actually is probably not worth writing since this pen is so hard to use. But this first part is called transcription, and the second part is called translation, and that's what we're going to learn more about now. And that is what the central dogma is. The central dogma is when you start off with this DNA, which can be replicated. You take this DNA, transcribe it into RNA, and then translate the RNA into protein. And that's the central dogma. So let's focus a little bit more on transcription, that first part. So we're still in the nucleus, and we're looking at our DNA. And what happens in transcription, and I really don't know why that's a K, must be from another country. Anyway, we take that DNA and open it a little bit, unzip it just a little bit. Because remember, like the cookbook, we don't want the whole cookbook, we just want a recipe, we just want a piece of that DNA. And it's not going to be exact copy, we are going to make a representation of that part of the DNA. What I mean by not an exact copy and just a representation is that the base pairing rules are slightly different. Instead of A's bonding with T's, when you are transcribing your DNA to RNA, your A's bond with a new base called uracil, which is a U. So as you can see down here, A bonds with U. Everything else is the same. T bonds with A, G bonds with C, but A bonds with U. C bonds with G, G bonds with C, G bonds with C, A bonds with U. So just keep that one slight difference in mind. And the other really cool thing is that once that little snippet is transcribed, so once you make RNA, or a representation of that piece of the DNA, then it separates from the DNA, like it's shown here, and the DNA winds back together. It's really cool. So then it's just like you have your cookbook and you write down the recipe on an index card and then you close your cookbook. So your cookbook remains the same. You didn't change the cookbook at all. You just took a representation of part of the cookbook and now you're going to use that for something else. So let's kind of practice a little bit about transcription. So just like we did with the replication, pretend that these are single strands of DNA. So not the ladder, not the two parts together, but just half of the DNA. And I'd like you to practice the base pairing rules, but instead of pairing with another strand of DNA, you're going to pair with RNA. So what I mean by that is instead of thymine, you will be using uracil. So A will bond with U. Remember, T still bonds with A. Everything else is still the same. C bonds with G. T bonds with A. G bonds with C. So everything is still the same except that A will bond with uracil, U. So for all three of these, practice the new base pairing rules. So you're starting off with DNA. Oops, that's supposed to be a D. Starting off with DNA. I can make that better. And you'll end up with RNA. Okay, take a second to practice that. So now that you kind of have the gist of transcription, we're going to move on to translation. Now, transcription's not going to happen by itself. I mean, it happens, but then it's used for translation. So it's separate, but the one is done before the other. So you have your DNA in the nucleus of the cell, and you take a representation of part of the DNA, and that's your RNA. That's what's done in transcription, what we just talked about. And that RNA is taken outside the nucleus, and it's taken to the ribosome. The ribosome is this blobby thing down here. Remember, the ribosome is like the chef. So the ribosome is going to read the RNA, and it's mRNA. It's going to read the mRNA. And the mRNA is going to tell it, call over these amino acids. And these funky shaped things are what carries amino acids over, and that's called tRNA, not super important. But these amino acids are these little black dots. So the mRNA tells the ribosome, call over specific amino acids in a specific order. And so it calls over, and it tells it that by sets of three bases. So like AUG would be a set of three. ACG would be a set of three, and each set of three is called a codon. So these codons represent one amino acid. Or not represent, but they are instructions for one amino acid. So the ribosome reads every three bases, or every codon, and every codon is basically telling the ribosome to call over a specific amino acid. And as those specific amino acids are called over to the ribosome, they bind together. So the amino acid bonds with the other amino acid, bonds with the other amino acid in a big long chain. And that big long chain ends up being your protein. So that's the bare basics of translation. And there's a little video that will be posted on the website if you would like to watch that. It kind of just again goes through an animation of translation that will be on the website for you to view if you'd like to. So let's just kind of wrap things up here, going through transcription and translation in order, taking DNA to RNA to amino acids that make a protein. Let me get my little pen here. So this is a two-sided DNA. So this is like a piece of DNA. As you can see, it follows the DNA base pairing rules: A with T, T with A, G with C, C with G, blah, 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 blah. So the first step, that transcription, that first step from DNA to RNA, that transcription step, means we're going to have to take this snippet of DNA and make it into RNA. So if we have this T, that is not going to be a T anymore. That's going to be a, or that's not going to, oh yeah it is. Oh, I just messed that up, sorry. The T will bond with A, but the A will bond with U. C will bond with G. T will bond with A, G will bond with C, C will bond with G, C will bond with G, T will bond with A, C with G, C, A. Now this is the difference here again, U instead of a T, and then two more A's. So that was transcription. Just did transcription. You took DNA and now it's represented in RNA. So that's where we stand now. Now, because we're not in the cell, we can't use the ribosome, we have to use this cheat sheet here. Ribosomes don't need, but we do, to take the RNA and then know what amino acids the RNA is going to call for to put together to make proteins. And so remember, 3 letters of the RNA are read at a time by the ribosome, and those are called codons. C-O, let's see if I can do this, D-O-N. So using this special chart, which you'll all have, and there's a copy of on the website, you can figure out what amino acid is represented by each codon. So AUG, you can see down here, AUG, it corresponds to an amino acid called methionine. And that's actually a really special amino acid. It's the amino acid that starts all proteins. It's called a start codon. So then we know that we're going to have a protein that starts with methionine, or MET is what it's abbreviated to. Then we're going to look at our second codon, ACG. So we look for A, for C, and for G. It's down here. And so that corresponds to the amino acid threonine. So that's T, H, R. Oh, that looks like an N. T, H, R. So now we have a sequence of methionine and threonine. Look at the next one, GAG. So G, A, G. And that represents glutamic acid, so GUL. Oops, GLU, slightly dyslexic, sorry about that. So as you can see, we're using these codons to gather our amino acids and put them together in a chain, and that chain represents our protein. So that's really the whole process of transcription, DNA to RNA, and translation, RNA to proteins. So that's really it, folks. I really like this. Oops. Oh, I lost it. I like this last picture. I think it's a really great graphic of the central dogma, where we start in the nucleus, where our DNA is stored, and we open it a little bit to make the mRNA, and then that mRNA is taken out into the ribosome, and then that mRNA instructs the ribosome to gather amino acids, and those amino acids are put together into protein. So I think this picture is really cool. Anyway, I hope that gives you some information on the central dogma and